Hello, everyone, and welcome to today's webinar, T-Cell Analysis, Fine-Tuning Your T-Cell Research Workflow Step by Step. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit miltenyibiotec.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. Our speaker will answer questions via the email address you provided during registration. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Marcello Stein, Global Marketing Manager, Translational Immunology, Miltenyi Biotec. Dr. Stein, you may now begin your presentation.
Welcome everybody to the third webinar of our T-cell series, T-cell analysis, fine-tuning your T-cell research workflow step by step. In the next 45 minutes, you'll learn about the best T-cell analysis products by Miltenyi Biotec. So enjoy. Please briefly acknowledge the displayed disclaimer. So let's begin with a brief company presentation. Well, Miltenyi Biotec was founded in 1989 by Stefan Miltenyi himself and is the largest owner-managed company in Germany. We have two main sites in Germany, one in Bergisch Gladbach, where the headquarters are located, and one in Teterow, where the GMP manufacturing facility is located. We have subsidiaries in 28 countries and distribute to an additional 73 countries. Currently, we employ over 4,000 employees worldwide. Our mission is to serve the needs in life science research and to translate new scientific findings into clinical applications. As for the agenda, first we will have a brief look at the biology and function of T cells, as well as at the concept of workflows and the hallmarks of MACS cell analysis. Next, we will have a look at our highlight T cell analysis products. We start with flow antibodies and kits, and then we will learn about our flow cytometry and imaging instruments. And finally, we will of course briefly sum up. Let's briefly introduce the T cells in general and of course our MACS flow analysis solutions. Well, T cells play as effectors of the immune system an integral role in the adaptive immunity. They can be subdivided broadly into several phenotypically and functionally distinct subsets, such as T helper cells, cytotoxic T killer cells, T regulatory cells, as well as the rather heterogenic group of unconventional T cells comprising, for example, natural killer T cells and gamma delta T cells. T cells are the only immune cells that are able to form a long-lasting immunological memory that helps to respond faster and more efficient to already experienced threats. Key surface markers that are always mentioned in regards to T cells are, of course, the T cell receptor TCR that recognizes antigens, as well as the activating co-receptors CD3 and CD28. Those three surface markers are present on all T cells. Furthermore, co-receptor CD4 and CD8 are the defining markers for T helper cells and cytotoxic T killer cells, respectively. To exert their functions as effectors of the immune system, T cells release cytokines to communicate with their environment and to cause various effects, such as promoting inflammation. These include interleukin-2, interleukin-4, interferon gamma, tumor necrosis factor alpha, transforming growth factor beta, and interleukin-10. T cells are most relevant in both maintenance of health as well as fighting diseases. Last but not least, these properties make T cells ideal tools for immunotherapy, where T cells, for example, are genetically modified as CAR T cells to attack cancer specifically. A quick look at T cell biology covers the basic aspects. When talking about T cell phenotypes and functions, mostly surface markers, cytokines, and transcription factors come into play as defining molecules that can be most efficiently analyzed, well, mainly by flow cytometry approaches. Well, although our best known products are indeed the microbeads and isolation kits, we are not just a cell separation company. We actually cover the whole experimental T-cell workflow from cell separation to cell analysis, the topic of today, including also sample preparation as well as cell culture. We provide thereby solutions for each step of your experiment. And today we concentrate on T-cell analysis. Please note we already covered T-cell separation and T-cell culture earlier this year in 2 different webinars available as recordings on our website. So our T-cell portfolio is indeed large. One way to keep a good overview is to divide our T-cell products into our different main workflow technologies. Sample preparation for everything that has to do with preparing your sample, for example, tissue or blood, before you isolate your T cells to start your experiment. The GentleMax tissue dissociator and the dissociation kits are here the hallmark products. Then we have cell separation, our classical microbeads and isolation kits in all their variants to isolate efficiently T cells and subsets. Cell culture, everything needed to culture, activate and expand your T cells from media to cytokines to antigens. And last but not least, cell analysis, T cell flow antibodies, flow kits, and also instrument for flow cytometry, sorting, and even imaging. Well, although it may be now easier to get an overlook of our product world, we thought that maybe the best way to arrange our large T-cell portfolio are distinct workflows, specifically designed for your experimental needs. Thereby, we cover as focus workflows, amongst others, regulatory T cells, CAR T cell research, gamma delta T cells, tumor-infiltrating leukocytes, polarization of T cells, antigen and virus-specific T cells, and of course, activation and expansion of T cells. With these comprehensive workflows, you can easily choose the products relevant for your specific field of interest. Please keep in mind, the T-cell portfolio is growing by the day, and the respective workflow overviews are all constantly evolving and work in progress. So check regularly for updates. By the way, you might notice throughout the presentation these small yellow buttons paired with a little click hand symbol, just like the one on the lower right. In the PDF version of this talk, you can access the respective product pages, marketing materials, videos, and protocols by simply clicking these buttons, as they are all hyperlinked. Well, getting your whole workflow from Miltenyi Biotec means ease of mind. Standardization, reproducibility, and organization of an experimental workflow become much more feasible. Miltenyi as supplier for the whole T-cell workflow means that all reagents fulfill the same high-quality criteria, and the order process can be streamlined. Thereby, harmonizing the steps of an experiment as well as automation and upscaling are easily performed. Getting all products from Miltenyi Biotec guarantees smooth procedures and less errors, since, for example, our cell separation is tested for compatibility with our flow cytometry and our media for compatibility with our cytokines and so forth. Moreover, having Miltenyi as full supplier for the whole workflow means also getting full support for every step of your experiment thanks to our great scientific support team. Moreover, a hallmark feature of Miltenyi's workflow solutions is the seamless translation from a research setting into a clinical setting and back, thanks to our Max GMP portfolio, as well as cell manufacturing platforms such as the Clinimax Prodigy. Well, when applying flow cytometry approaches for T-cell analysis, of course, precision is key. However, unwanted variability in analysis results can have many different sources, such as donor variability, inconsistent panel design, reagents unspecificity, unprecise instrumentation, and operator and analysis variability. Here you see just a small example of one of our publications in the Journal of Human Gene Therapy. Immune cell composition greatly varies between donors depending on possible disease states and starting material preparation. All these sources of variability should ideally be taken into account and compensated for most efficient flow analysis. That is why max flow cytometry stands for highest precision and covers all aspects of flow cytometry. Flow analyzers, flow sorter, imaging systems, and all the respective reagents are included. And this shows in the width of our and diversity of our flow portfolio, not only for T-cell analysis, from instruments to software, from antibodies to flow kits, and even customization solutions. We offer indeed the whole range of flow analysis. So let's start with our highlight flow products, our flow antibodies and flow reagent kits. First, we have the REAfinity Recombinant Antibodies. Being recombinantly engineered, we have full control over their genetic sequence and can express the REAfinity recombinant antibodies in mammalian non-hybridoma cell systems. This means that REAfinity antibodies exhibit highest purity and lot-to-lot consistency for greater reproducibility, that they can undergo a most stringent in-house validation and have no need for tedious and costly Fc receptor blocking. And they require only one universal isotype control for maximum convenience and cost saving. Most recently, even releasable versions for label-free cell sorting were added to our REAfinity portfolio. Additionally, REAfinity antibodies also are part of our unique Miltenyi Biotec tested panels, various detection kits, and pre-mixed cocktails, including the innovative Stain Express dried antibody cocktails. REAfinity antibodies are hereby readily available in hundreds of specificities and are best combined with our proprietary Biodye fluorescent antibody conjugates. Thereby, our Biodyes are bright, with superior mean fluorescence intensity for excellent signals, distinct with a high stain index for clear population discrimination, and hassle-free, since they are ideal for multicolor experiments due to low compensation. All this makes REAfinity recombinant antibodies the ideal choice for flow analysis, since traditional hybridomas are not standardized in nature. More info on that can be found following the various links on this slide. And indeed, if comparing classical hybridomas to recombinant REAfinity antibodies, it shows that without FcR blocking, hybridomas bind unspecifically, leading to an overestimation of the actual cell frequencies. REAfinity antibodies instead do not need any FcR blocking and always bind precisely, leading to lower background and reproducible results. Well, and now when comparing also antibody preparations of classical hybridomas to our REAfinity antibodies, in this case, it is all mouse CD4 monoclonal antibodies analyzed by mass spectroscopy, it becomes obvious that the classical hybridomas contain a large amount of non-functional contaminating light chains, while REAfinity antibodies are without these contaminations. This difference comes from the recombinant nature and preparation of REAfinity antibodies and the pure mammalian expression system. As for the Biodyes, our proprietary fluorophores also bear really some nice advantages. When comparing directly fluorescence intensities of dyes with similar spectra, Biodyes generally are superior, as shown here in the comparison between Horizon V500, Chrome Orange, and our Biogreen. Furthermore, Biodyes come in many varieties, as shown here in this brief example. By combining them with classical fluorophores, the possibilities are really endless. Here, I'd like briefly to show just some exemplary flow antibodies that you can choose from to create your own panel, depending in which context you want to analyze your T cells, be it in development, activation, exhaustion, or any other type of phenotyping. Please note that the relevant markers may differ, of course, between the species, and the panel should be adapted accordingly. However, to ease things up, we also offer our Miltenyi Biotec tested panels, as well as our convenient antibody panel builder, which is a web-based software tool that you can access to build your own panel. Just follow here again the links below to have a look. Apart from classical antibodies and flow panels, we also offer a wide array of flow analysis kits, containing, of course, our proven antibodies to analyze T cells in really every way, namely for the analysis of cytokines, for phenotyping, and of course, viability. These flow kits and assays, but also our tested panels, cover applications and parameters to assess such parameters as purity, for example, after enrichment, viability, the phenotype, of course, the immune cell composition of your sample, the activation and exhaustion status of your T cells, the transduction efficiency, the potency of, for example, your killing CAR T cells, and many, many more. So let's have a look at the first three of our convenient, ready-to-use flow analysis kits, such as first the Rapid Cytokine Inspector kits, as all-in-one no-worry solution that combines surface marker staining with intracellular cytokine staining for the analysis of activated CD4 and CD8 T cells in the fastest way possible. Second, the MACSQuant cytokine kits for determining concentrations of up to 12 different secreted cytokines in cell culture supernatants. And third, the 8-color immunophenotyping kits for flow evaluation of various leukocyte subsets, for example, monocytes, neutrophils, eosinophils, and T, B, and NK cells in your sample. Importantly, all of these kits are compatible with the fast and convenient express modes on the MACSQuant analyzers. These software modules automate analysis and thereby not only speed it up, but also eliminate a human error thanks to automated gating and analysis templates. But later we will hear more about that. Make sure to have a look at the new promo video for the flow kits as well as the express modes and the MACSPlex webinars by following the respective links on this slide. Consequently, I'd like to present some applications examples using the three aforementioned kits. Well, the Rapid Cytokine Inspector is the ideal solution for the analysis of rare antigen-activated T cells. It combines extra and intracellular staining of subtype and activation-specific surface markers and cytokines at the same time. In this example, T cells within the PBMC context were stimulated with CMV peptide pools, so our peptivators, PP65 and immediate early, and the frequency of CMV reactive CD4 and CD8 T cells were analyzed by assessing interferon gamma expression. Hereby, classical intracellular staining with the handmade protocol and our Rapid Cytokine Inspector kits were compared. And as you can see, the Rapid Cytokine Inspector performed at least as good as the classical intracellular staining, but yields the benefit of a much faster protocol and less washing, less cell handling, and then of course less cell loss. Hereby, a classical intracellular staining can take up to hours, while the Rapid Cytokine Inspector, with the Rapid Cytokine Inspector, you're done in only 45 minutes. Further applications include assessment of a sample's immune status or testing of leukapheresis material for virus-specific CD4 and CD8 T cells. As said before, the Rapid Cytokine Inspector is of course compatible with the Express mode for automated data and analysis on the MACSQuant analyzers to even speed up your analysis more. Also, in the context of CAR T cell analytics, the Rapid Cytokine Inspectors come in quite handy, as the readout is as straightforward as it gets. Here in this example, CAR T cells were co-cultured with target cells, either expressing the intended CD19 antigen or not. As you can see, CD3 positive T cells, and alternatively, you can with the Rapid Cytokine Inspector kits also differentiate between CD4 and CD8 positive T cells, were assessed for interferon gamma, TNF alpha, and interleukin 2 intracellular cytokine expression. Co-culture of CAR T cells with antigen-negative target cells did not lead to a proper T cell activation with no or only low cytokine expression. Whereas co-culture of CAR T cells and antigen-positive target cells, as you see here, led to strong expression of interferon gamma, TNF alpha, and interleukin 2. Our next application examples involve the MACSPlex cytokine kits. So the second kit that I presented before. Also here, CAR T cells were co-cultured with target cells expressing the respective target antigen, in this case CD20. In this example, we evaluated the functionality and antigen-specific killing of manufactured CAR T cells using the MACSPlex assays. In more detail, we co-cultured the CD20 transgenic positive target cells, in this case it was Raji cells, with our CD20 directed CAR T cells, be it from healthy donors or cancer patients, and measured cytokine secretion. And as shown here, upon direct contact with the target cells, the CD20 directed CAR T cells secrete high amounts of effector cytokines like interferon gamma, interleukin 2, and TNF alpha, verifying their functionality. In the next example, we will have an application example of the 8-color Immunophenotyping Kit, the third kit that I presented before. Here, a single cell suspension of a dissociated tumor was stained with the 8-color Immunophenotyping Kit and analyzed by flow cytometry using the MACSQuant Analyzer to determine the immune cell composition in the dissociated tumor sample. The tumor was first dissociated with the GentleMax tissue dissociator and the respective enzyme kits. Then the suspension was stained with the 8-color Immunophenotyping Kit human. And based on the predefined displayed gating strategy using the express modes, you can precisely identify the amount of total leukocytes, as well as the CD8 positive, the CD4 positive T cells, the NK cells, the NKT cells, B cells, CD14 plus monocytes, subsets, and so forth. But of course, we have more kits to offer when it comes to flow cytometry. So apart from the 3 aforementioned flow kits, we also offer further interesting T-cell analysis kits in our flow portfolio. One of our more recent highlight flow analysis kits is depicted here, the SARS-CoV-2 T-cell analysis kits human. These all-in-one kits allow the efficient stimulation and sensitive analysis of SARS-CoV-2 reactive T cells in human PBMC or whole blood samples. After stimulation with SARS-CoV-2 peptidated peptide pools, cells are stained with our optimized flow panel, as depicted here on the upper right, and analyzed, for example, on a MACSQuant flow analyzer. Depending on the version of the kits hereby include a SARS-CoV-2 peptidator of choice, as well as the whole SARS-CoV-2 T-cell flow panel consisting of 8 to 9 REAFinity Recombinant Antibodies. Moreover, all needed buffers and controls are included, such as the positive control cytostim and, if needed, the live dead marker viability fixable dye. As you can see, we can beautifully detect, for example, interferon gamma, TNF alpha, and CD154 expressing SARS-CoV-2 reactive CD4 and CD8 positive T cells after stimulation with a mix of SARS-CoV-2, protein S, protein N, and protein M peptidators. Of course, also the other markers as depicted here in the panel can be easily assessed on the flow cytometer. Please keep in mind, although the kits are called SARS-CoV-2 T-cell analysis kits, we have also a version without peptidators that is perfectly suited to analyze any kind of antigen-reactive T cells, as you are here free to choose any stimulus of choice. In the following nice datasets, we stimulated PBMCs of SARS-CoV-2 reactive donors with the aforementioned SARS-CoV-2 peptidator peptide pools as indicated. A mix of CMV and EBV peptidators was used as positive control. And as you can see, we pre-gated then either on CD4 or CD8 positive T cells and assessed CD154 and TNF alpha expression, as well as CD69 and interferon gamma expression, respectively, on the MACSQuant Analyzer. The reactive donors T cells respond specifically to the stimulation in comparison to the negative control, with the expression of the respective activation markers and cytokines. Moreover, I want to point out a cell analysis and separation product that is absolutely unique, the Cytokine Secretion Assays. They allow to detect and enrich viable cytokine secreting cells from mouse and human samples. Cytokines are immobilized on the surface of the secreting cells by the so-called catch reagent. Basically, thereby the cell labeled itself with its own cytokines. In a second step, the immobilized cytokines are then labeled with fluorophore-conjugated antibodies called detection reagent and can then be detected viably without intracellular staining on any flow cytometer. In an optional third step, the fluorophore-labeled cytokine-secreting cells can be specifically enriched and used in downstream assays by using anti-fluorophore magnetic beads. So consequently, our cytokine secretion assays come in two versions. For flow detection only, we have the so-called detection kits. For the separation of viable cytokine secreting cells, we have the detection and enrichment kits, including the additional enrichment steps via a MACS column. Please see in this application example of the IL-17 cytokine secretion assay detection and enrichment kits, how nicely antigen-specifically stimulated T cells can be enriched even from low frequencies to clearly visible levels above background, solely based on their IL-17 secretion. Antigen-specific T cells were stimulated within the PBMC context with a C. albicans peptivator for 16 hours or left untreated, and then enriched using the IL-17 secretion assay cell enrichment and detection kit. Before enrichment, with the cytokine secretion assay kits, levels of reactive T cells, be it in the unstimulated control or in the stimulated samples, are hardly visible. We are talking about really, really rare cells. However, after enrichment, analysis is much clearer. Now, when gating on the CD4 positive T cells and looking specifically at CD154 expression, it becomes apparent that mainly the CD4 plus CD154 plus T cells express in this example interleukin 17. Also in our second example, the cytokine secretion assays detection and enrichment kits work very efficiently also to enrich virus-specific T cells based on their cytokine secretion. Here, PBMCs of a CMV positive donor were stimulated for 4 hours with CMV lysate. The responding cells were stained and isolated according to the secretion of TNF-alpha and interferon gamma using the respective secretion assay, cell enrichment and detection kit. And as you can see, in the stimulated samples, a strong population of cytokine secreting CD4 positive and CD8 positive T cells respectively was enriched. In the unstimulated control sample, however, no cytokine secreting T cells were detected after enrichment. Moreover, in T cell analysis, of course, also analysis of cell viability plays an important role in a variety of contexts. Also here, we offer quite an array of different solutions, such as the viability fixable dyes, Annexin 5 kits, the 7-AAD staining solution, of course, propylium iodide, live dead cell discrimination kits, as well as staining solutions such as Höchst, DAPI, DRAC5, and Cytrac Orange. Again, you can directly access the respective product pages using the links on this slide below. Viability staining can, for example, be used to monitor cell fitness in regards to their storing conditions. In this example, for manufactured CAR T cells, first storage at 4 degrees for 48 hours, and second, freezing thawing, both with subsequent culturing at 37 degrees, were compared. We observed that cell fitness, basically the amount of apoptotic as well as dead cells, was higher after the freezing thawing cycle than just storage at 4 degrees. That way, your manufacturing process can be coordinated in regards to your needs and be, of course, optimized. In another extraordinary application example of viability stainings, CAR T cells mediated target cell killing was assessed via in vitro killing assays. CAR T cells and mock transduced T cells were co-cultured with antigen positive and antigen negative target cells. In this example, the target antigen was CD20. And of course, different ratios between CAR T cells and target cells in our Tex-Mex T cell medium. After co-culture and incubation at 37 degrees, propedium iodide staining of the sample was performed automatically by our MACSQuant flow cytometer just before sample acquisition, and the PI positive cells were measured. The measuring was then performed using a specific express mode, which is an automated and standardized analysis software exclusive to our MACSQuant analyzers. The percentage of killed, so propedium iodide positive target cells, is normalized to the control setups, for example, the MOX transduced cells or the target antigen negative cells. And as you can see in the data on the right here, killing of target cells amounted to about 40% at a 1-to-1 ratio, be it from CAR T cells obtained from healthy donors or patients. Our flow kits are rounded off with our outstanding CAR detection reagents. These reagents were developed to stain and detect transgenic T cells expressing CD19 CARs on the cell surface. The reagents come ready to use and with a muted IgG1 Fc for minimal background staining so that no FCR blocking is needed. The binding thereby of the reagent to the CAR T cells simulates the in vivo binding of the CAR to the respective tumor antigen. CAR-positive T cells can hereby be easily detected with highest precision. In this example, CD19-positive CAR T cells made up around 20% of the whole CD3-positive population in the samples. Of course, our CAR detection reagents come also in additional specificities, such as CD22, BCMA, or CD33. The next group of T cell analysis highlights products are our flow cytometers, sorters, and imaging instruments. Namely, our flow analysis and sorting instruments of the MACSQuant series. First, we have the MACSQuant Analyzer, powerful benchtop flow cytometers with up to 16 optical parameters. The clue here is that they allow for full automated acquisition using a robotic sample collector with the Express Mode software modules. Even data analysis is then automated and free of human error. The small benchtop cytometers fit easily under every flow hood, adding further to operator safety, for example, when working with contaminated samples. Second, we have the MACSQuant Taito cell sorter. Opposed to classical droplet sorters, the MACSQuant Taito allows for the most gentle sorting and highest viability even for rare and fragile cells, thanks to its unique microchip sorting technology. Furthermore, the closed and sterile cartridge system makes the Taito the ideal machine for a GMP environment. Well, a little bit more in detail, the MACSQuant Analyzer are not only small, convenient, powerful, but also offer a high degree of automation for startup, shutdown and cleaning, sample labeling and processing, calibration and QC, fully automated volumetric cell counting, and of course data analysis with the express modes. This makes them simple to use even for unexperienced flow beginners. Last but not least, with our support and the 21 CFR Part 11 implementation, you have every experiment under full control. So what are actually these aforementioned express modes exactly? Well, the express modes are software modules on the MACSQuant analyzers that automate flow analysis with standardized algorithm-based data evaluation. Results become more standardized as operator variability is minimized due to a lack of manual intervention and reduction of human error. This results in highest reproducibility with identical flow analysis independent from the operator. Express modes are simple to use and offer a quick and robust analysis for easy integration into SOPs with the validated automated analysis concept. Several of our flow analysis kits, such as the aforementioned Rapid Cytokine Inspectors, the MACSQuant Cytokine Kits, the 8-color Immunophenotyping Kit, as well as very specific flow panels, for example for CAR T-cell analysis, are compatible with the express modes, and your analysis will be automated. Please have a look at the respective webinar by following the link below. As just mentioned, we have, apart from cytokine and phenotyping flow kits, also a variety of CAR T-cell REAfinity flow panels that are compatible with the automated express mode analysis to standardize your CAR T-cell experiments. Thereby, we offer, for example, tested panels amongst others for CAR T-cell transduction efficiency, CAR T-cell stem cell memory phenotyping, CAR T-cell differentiation, CAR T-cell persistence, CAR T-cell proliferative ability, CAR T-cell activation, and finally, as highlighted here as an example, CAR T-cell exhaustion. After the MACSQuant Analyzer flow cytometers, I'd like also to have a closer look at our MACSQuant Titer cell sorter, the gentleman under the cell sorters. With its unique capability, namely the most gentle sorting in the closed sterile cartridge system, the Titer is ideal for a GMP clinical environment, not only in the fields of CAR T cells and Tregs, but also for antigen virus-specific T cells, including also tumor-infiltrating leukocytes. So let me point out that the Titer is already part of several clinical cancer trials involving antigen-specific, or in this case, better said, tumor-specific T cells. Respective data and testimonials can be found, of course, on our website. Worthwhile mentioning is, of course, also the Titer safety in virus research. The Titer works without internal fluidics and applies a sterile and closed cartridge system for maximum operator safety. Sorting purity is, of course, guarantees, as you can see here in the example below, where CAR T cells were sorted after expansion and staining with our CAR detection reagent to over 99% purity. On top of all that, a really fantastic novelty in our portfolio are our new imaging systems, MACSima and the ultramicroscope light sheet microscopes. These systems allow the visualization of T cells and others within a tissue context. Thereby, for example, new CAR T cell targets may be identified, the distribution dynamics of tumor-infiltrating leukocytes in tumor tissue assessed, and the deep phenotypic analysis of patient samples and the tumor microenvironment is possible. Hereby, the MACSima imaging platform for ultra-high content imaging applies the renowned MIX technology. Spatial immune cell distribution, including their phenotype, is assessed within tissue and cell conglomerates by the sequential staining of hundreds of markers. In contrast, traditional staining techniques allow maybe the staining of 3 to 6 markers, giving only an incomplete picture. This analysis of hundreds of markers within a single sample includes, of course, automated sample processing and strong software analysis and imaging of all types of fixed samples from tissue to single cells. Have a look at the product video by clicking the respective links on the left. On the other hand, our light sheet microscope of the ultramicroscope family enable you to visualize whole biological systems in 3D. Moving the samples through the light sheets allows the generation of sharp and bright 3D images of, for example, the tumor microenvironment, thereby really giving you the full insight of the tumor immune cell interaction. In this context, the ultramicroscope 2 is a highly flexible light sheet microscope for 3D imaging of cleared samples. The ultramicroscope Blaze is a fully automated light sheet microscope for 3D imaging of multiple samples. Please have a look at this impressive video taken with the ultramicroscope Blaze that shows a mouse xenograft of a human pancreatic carcinoma cell line with infiltrating CAR T cells in 3D. And here we come to an end of this presentation, and therefore I'd like to briefly sum up. As mentioned before, the variety of our T-cell product is best organized in comprehensive workflows following your scientific interest. Consequently, please find here listed our highlight application workflows with clickable links to the respective application and protocol pages, as well as to further info material and webinars. Of course, there are more and our selection is growing by the day. However, our current highlights include polarization of T cells with protocol and cytokine combinations for the most efficient polarization of your CD4 positive T helper cells. The regulatory T cell workflow with all needed solutions for the isolation as well as the expansion and of course flow analysis of rare Tregs in a translational setting. The CAR T cell research workflow, covering all steps from T cell isolation to activation to transduction and finally CAR T potency analysis for fully translational and functional generation of CAR T cells in a research or small scale setting. The tumor-infiltrating leukocyte workflow to efficiently dissociate tumor tissue and isolate even the rarest tumor-infiltrating leukocyte. The antigen-specific T cell workflow, optimized for the specific stimulation of antigen-reactive T cells as well as the isolation and analysis of these extremely rare cells in many different contexts. And finally, the COVID-19 virus-specific T cell workflow that covers each step from stimulation to enrichment to flow analysis of SARS-CoV-2 reactive T cells, especially important during the current pandemic. Have a look at the respective application pages and webinars to find out more. And of course, also here we will update and expand the highlight workflows regularly. So please have a look for updates. Although that was quite an extensive overview of the T cell portfolio, you have just to remember 3 easy things. Well, first, we offer more than just T cell separation. Second, we cover the whole T cell workflow. And third, we offer a solution for every pain point in your experiment. So have all the success with our innovative and complete T cell portfolio as displayed here in this full-featured overview. Again, please keep in mind the T cell portfolio is growing by the day and the respective workflow overviews are all work in progress. So check back regularly for updates. Please also follow these links here on the lower right to access the respective T cell application product pages as well as to download our T cell brochures. Talking about more information and brochures, of course, you can find a variety of helpful marketing material downloadable from our webpage, www.miltenyibiotec.com. For T cell specific applications, just click on the button on the lower right. You'll find flyers and brochures, webinars and videos, protocols and datasheets, customer reports, application notes, scientific posters and datasets. And of course, you can also directly always reach out to the responsible sales representative in your area. And hereby, I end this presentation with this nice picture of our company campus in Bergisch Gladbach at night. So thank you very much for your attention.
Thank you, Dr. Stein, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Questions will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.